Thank you. Thank you, Derek, and welcome. I'm looking forward to having a chance to present my talk on bugs in the belly, the good, the bad, and the ugly. Now, the first thing I have to tell you is that it's really not about bugs in the belly. It's actually bugs in our intestines. And the first question I want to ask you is, why do we need to understand them? Well, you probably already know that bugs in our intestine promote gut health. You may know that bugs in our intestines have now been linked with obesity. What is also interesting is that bugs in our intestine play a significant role in disease. Some of the diseases like type 1 diabetes and inflammatory bowel disease, among others that we're going to talk about today. You may also be aware that they are the cause of foodborne illnesses. And this is a perfect talk for a lunchtime. And then finally, we now are beginning to understand how we manipulate bugs to promote better gut health. So let's now take a look at the good bugs. And that is what we're going to talk about as the human gut microbiome. So in fact, living in our intestines right now is between 10 and 100 trillion microorganisms. Now, I want you to think about that for a minute. In fact, it would take you 13,000 years to count to 1 trillion. But we're not talking about 1 trillion, we're talking about 100 trillion. And in fact, the bugs in your intestines actually outnumber the human cells in your entire body by 10 to 1. So there's a lot of them. There are over 500 to 1,000 different species of bugs living in your intestines, and collectively we call them the gut microbiome. What do they do for our health, this gut health that we're talking about? Well, they help us process nutrients. In fact, some of the nutrients that you're consuming now. They help us harvest energy from foods so that you can run faster. They help provide protection against pathogens, the bad guys, and they help develop our immune system. Okay, so let's ask the question, who are they and where are they? Well, as I mentioned before, there are between 500 and 1,000 different species living in your intestines, and the numbers of bacteria do increase as you progress through your GI tract. And in fact, we have fairly large numbers in the small and large intestine. So if you take a look here, nice cross-section of our GI tract with the esophagus to begin with, stomach, and then leading into the small intestine and the large intestine. Now, I just want you to think for a minute about how long your small intestine is. In actual fact, your small intestine, if we stretched it out, in the average adult, is between 20 and 23 feet long. It's pretty long to be packed into here. Secondly, our large intestine, although you might think the name suggests that it's longer, is not. It's larger in diameter, but it's actually much shorter. It's around 5 feet long. Nevertheless, between the small and the large intestine, there are lots of places for bugs to hang out. Okay, let's talk now about the composition of the gut microbiome along the GI tract. And we're going to start here with the duodenum and go all the way to the colon. In other words, starting at the small intestine and going to the end of the large intestine. In fact, what we find is that there are different species of bugs living in various sections of our small and large intestine, and the numbers of species increase as we go from the small to large intestine. Okay, how do we acquire our gut microbiome? Well, in fact, you acquire it at birth, mainly from your mother. Studies are now showing that we acquire our gut microbiome, and we acquire it differently depending on the birth delivery method. In other words, if you've been born by C-section or not, it will differ. Whether you've been bottle-fed or breastfed, it will differ. And I have to say, these studies are brand new. In fact, the publication just came out in January 2014. We're just beginning to understand how this affects gut health. What are the long-term consequences of the difference between being born by C-section and your gut microbiome? We don't know yet. We know that your gut microbiome develops over your lifetime, but it develops up to the age of 2, and then it's fairly stable until you reach about old age, which according to these studies has been defined as 65. Okay, so what happens if your gut microbiome is disrupted? Well, let's take a look. Let's start out with a cross-section of normal gut cells. So here are your normal gut cells, and living along the layer of your normal gut cells is your gut microbiome. But what happens if it becomes disrupted? Okay? And we're going to talk about various things that can disrupt your gut microbiome. In fact, it turns out that disruptions in your gut microbiome have been linked with a number of different infections and diseases, such as autism, inflammatory bowel disease, chronic fatigue syndrome, insulin resistance, and diabetes, obesity. And in fact, this list is growing as we continue to do our research and understand more about how the gut microbiome plays a role in various diseases and in our health. So let's now talk about the link between the gut microbiome and obesity. And for this, scientists have done a lot of studies on obese mice and lean mice. The first thing they find is that bacterial communities in obese mice are not the same as bacterial communities in the lean mice. The second thing that they find, and I bet you this doesn't surprise you, is that the bacteria from the obese mice are better energy extractors. And that kind of makes sense, doesn't it? Because they're better able to extract energy from our foods, so of course they're going to support more fat storage for their host. But this is the most striking part of the research that's been done on these mice. When they take bacteria from the ceca, that's a portion of the intestines, when they take bacteria from the obese mice and they feed them to the lean mice, here's the astounding part. The lean mice gained weight over a 2-week period despite being on the same diet and the same food intake. Okay, well, let's ask the question now. How does this translate to humans? How is the gut microbiome different in obese and lean people? And for this, they've done studies with what are called discordant twins. And discordant twins are twins where one twin is 10% or more heavier than the other twin. And in fact, what they found is, and you'll see here, they found a whole lot of differences in the obese twin, and we're not surprised about some of these differences, like, for example, the signal to tell the brain that you're satiated or full decreases in the obese twin, and so on. Changes in the liver, changes in the ability to store fat in adipose tissue, changes in muscle, and changes in epithelia. But in fact, what we're interested in today is the fact that the gut microbiome of the obese twins shows altered composition and also, like in the mice, showed an increased ability to harvest energy. Okay, let's now go and talk a little bit more about the link between the gut microbiome and brain function and behavior. So in fact, there have been a large number of studies done to compare autistic and neurotypical children. And what they were looking at with respect to these studies was how does the diversity of the gut microbiome in autistic children compare with those in neurotypical children. And you can see here the autistic children are in the red, so you can see these graphs show you that there is a lower level of diversity in the autistic children generally, and you can see with the blue lines they're higher, and that suggests that there is a higher level of diversity in the neurotypical children. And that's summed up with this bar graph that we're showing here. So what they found is that there is a decreased diversity of the gut microbiome, and that appears to be linked to an increased risk of autism. When they take a look at other studies and how that affects behavior, they have found that when you have a disrupted intestinal gut microbiome, it seems to be associated with alterations in behavior, in cognition, and emotion. And of course, these studies are ongoing. We still don't understand exactly how this plays a role, but we know it does. We know there are links. Okay, let's now take a look at the gut microbiome and inflammatory bowel disease. And here we have a picture of a healthy colon, if you were looking straight down the colon. And here we have a picture of an ulcerative colon from an individual who has inflammatory bowel disease. And you can see that it's much redder, inflamed, looks painful, and it is. They found that in these studies, they found the gut microbiome likely plays a key role in inflammatory bowel disease. They've also found that the gut microbiome appears to trigger an inappropriate immune response. In other words, for some reason, it's alarming the immune response in those individuals. They also found that there's a change in the composition or in the numbers of the gut microbiome in the gut, and it appears to predispose these individuals to infection. And so, of course, that leads me now to talk about a potential role in inflammatory bowel disease for pathogens, or what I'm going to call the bad guys. Okay, so let's take a look now at some of the bad bugs. So these are bugs that cause a variety of diarrheal diseases. Now, there's a whole group of diarrhegenic E. coli that cause diarrhea disease: Salmonella typhimurium, Campylobacter jejuni, Shigella flexneri, Listeria monocytogenes, and Yersinia pestis, to name a few. But what I'm going to focus on for today is the diarrhegenic E. coli, because that's in fact actually what I do my research on. Okay, so let's take a look now, because we're going to look really at these ugly bugs. Two species of bugs that I'm particularly interested in are something called the Shiga toxin-producing E. coli, and I'm going to nickname them STEC, and enteropathogenic E. coli, and I'm going to nickname them EPEC. So, Shiga toxin-producing E. coli is responsible for causing acute and sometimes fatal diarrheal disease. Now, you may have heard about the outbreak in Walkerton. That was in the year 2000. So you probably would have heard that from your parents. But if you did hear about it, it was a huge outbreak of diarrheal disease associated with STEC, and it actually resulted through contaminated drinking water. And in fact, it sickened over 1,000 people and caused several fatalities. You may have also heard that eating undercooked hamburger could give you diarrheal disease. In fact, the diarrheal disease that we're talking about is STEC. What happens if you consume undercooked hamburger or you drink water that is contaminated with STEC? 3 to 5 days later, you begin to experience diarrhea. The diarrhea turns bloody. That's a sure sign that you've got STEC, or at least it's a potential contaminant that you've consumed. And in fact, if you have bloody diarrhea, that's definitely the time to go and seek medical intervention. For most people, that diarrheal disease clears in about 10 days. You are miserable for 10 days. You have vomiting, you have severe bloody diarrhea, you really are very miserable. But for most people, it does resolve. However, there is a percentage of people, generally young people or elderly, who become quite ill with what we refer to as systemic complications. And that's because the toxin that the bug produces actually gets into your systemic circulation and can cause kidney failure, spleen and liver damage, and even brain damage. So it's very serious, and of course can be, as I mentioned before, fatal. it leads to something called hemolytic uremic syndrome, which is in fact essentially renal failure. Okay, let's now talk about enteropathogenic E. coli. So it is also an important and deadly cause of diarrheal infection, but particularly in infants and small children. And we find EPEC more frequently in the developing world, although we are now seeing it in nursery schools, daycare centers, worldwide. And so this is a significant concern. It causes chronic and sometimes fatal watery diarrhea. So it's another very important pathogen in terms of understanding its mechanism. So let's now talk about what these bad bugs actually do. So when you ingest them, they make their way down to your small and large intestines, where they bind to our intestinal cells. They then actually produce molecular syringes, and they're very cool to see these molecular syringes. And they use these molecular syringes to inject proteins into our cells that disrupt our cells' function. They also hijack our normal cells' biochemical signaling. So within all of our cells, there's a whole lot of biochemical signaling that's going on that is part of our normal cell function. The bugs disrupt our cells' biochemical signaling. The other thing they do is they evade our immune system. So they actually evade detection from our immune system, which allows them to multiply. Some of them, as I mentioned before, secrete deadly toxins. And those are the toxins that are responsible for causing the systemic systemic complications that I mentioned earlier. Okay. And in fact, taking a look at this picture, you might ask, what on earth is this? Well, this is actually EPEC, one of those pathogens that I mentioned to you earlier. This is this purple bug sitting on top of a section of your host cells. In fact, what we find is when the bugs actually bind to your intestinal cells, the host cells actually rise up and form this strange-looking pedestal structure. And this is typical of both STEC and EPEC infection. So the question is now, what makes these bugs so virulent? And that leads me into the research that I'm doing here at Ryerson. What I'm going to do now is take you on the bad bugs journey through our GI tract. So for that, we need a cross-section of our GI tract. So let's talk about it. You eat that hamburger contaminated with STEC. Oh, and by the way, if you think you need a lot of STEC in the hamburger to make you sick, you're wrong. Between 10 and 100 colony-forming units, 10 and 100 will make you ill. So it's very little STEC that is required to cause infection. So the first thing that's going to happen is you're going to swallow it, and it's going to end up in your stomach. Of course, you know that the stomach, there's lots of gastric acid there, and for those of you that have already studied the term pH, the pH is as low as 2.5. Depending on how long the food stays in your stomach, that bug could experience that very low pH for up to 2.5 hours. That's a long time to hang around the stomach. So part of my research is to ask, how does that affect the bug? Second thing is, then the bug passes from your stomach into your small intestine, where it encounters bile. Bile, as you may know, its function is to emulsify fats. It has a detergent action, and that actually disrupts the bacterial membrane. So it's not a pleasant experience for these bugs either. So again, I want to ask the question, how does that affect them? Okay, then the bug enters the large intestine, or the colon. And there it encounters more stressful situations. First thing is something called short-chain fatty acids, which are stressful for the bacteria. Something else that it encounters are low oxygen concentrations. Again, stressful. And thirdly, competition from normal gut microbiota. They have to compete with your healthy gut microbiota in order to be able to colonize and infect you. Okay, so let's now think about the fact that even though they have had to pass through the stomach, they have had to evade the bile or pass through and cope with the bile and then enter the large intestine and deal with those stresses, they still manage to go on to cause infection. So the question that I pose in my research is, could they be using these stresses like signposts to tell them where they are on their journey? So let's now talk about some of the things that we've found in my research. First of all, the impact of acid. So here we are looking at the stomach. What we've in fact used is something called a DNA microarray to understand how acid affects STEC. And what we found when we did this is, we found that there was the bug, after experiencing acid like it might experience in passing through your stomach, showed improved ability to resist acid. It showed an increased ability to repair DNA. And it also produced more flagella. Now, flagella are these long finger-like projections that the bacteria use in order to move. So, of course, what that suggested to us then is that STEC may actually be using the acid as a cue to arm itself to do three things. One, to resist the acid. Two, to repair the DNA damage that is caused by the acid. And three, to swim away from the acid. Going on now to the impact of bile, we did a similar study using DNA microarray. And what we found is that bile causes the changes in the bacterial coat. And in fact, what it specifically does is it alters the charge of the bacterial coat, making the bacterial coat less negative. And of course, our question was, why? What advantage would this provide for the bacteria? Well, interestingly enough, there are some molecules called defensins that are produced by specialized human small intestinal cells. And these defensins are actually antibacterial molecules that hunt out the bacteria and kill the bacteria. And they hunt the bacteria out by recognizing the negative charge on their coat. Well, of course, if the bacteria has reduced the negative charge on its coat, the bacteria are no longer going to be detectable by the defensins. So what we saw from this is that STEC may actually be using bile as a cue to put on an invisibility cloak to escape killing by the defensins, much like our friend Harry Potter put on an invisibility cloak. Okay, let's talk about the impact of short-chain fatty acids now. As I mentioned to you before, there are short-chain fatty acids in the small and large intestine, and in fact what we find is that there are increasing levels of these molecules as you go from the small to the large intestine. When we did another DNA microarray study, what we found was that the bugs produce more flagella under low concentrations of short-chain fatty acids and fewer flagella under high concentrations. And again, we asked the question, why? What advantage could that serve the bug? Well, in fact, STEC actually colonizes or binds to our cells in the large intestine, so we're down here. So it looks like STEC may actually be using the changing levels of short-chain fatty acids to alter its motility. So in this section of the GI tract, it will want to swim. In this section, it will want to slow down and attach and bind. So in fact, more flagella here will insist it to swim, Fewer flagella here are required because now it's slowing down, stopping, and binding. So this, in fact, we think is part of a strategy for allowing the bugs to know when to stop and bind. So from our results, we are finding that STEC may in fact be using these cues that are found in the microenvironments of the GI tract to both optimize survival in the case of the acid studies that we've done, and also to optimize infection strategies, as we saw in the case of bile and short-chain fatty acids. Now what I'm going to do is I'm going to take you back to the beginning, and we're going to talk a little bit more about that gut microbiome, the good bugs. I've mentioned to you already that the gut microbiome can be altered by a variety of factors, including diet, antibiotics, stress, infection, and even genetic factors. We also have talked about the fact that disruptions in our gut microbiome may increase the risk of disease and infection. The next question for us then is there any way to restore the healthy gut microbiome? Well, in fact, research is showing that there are a variety of ways, although it's still in its infancy in terms of understanding this. There have been a lot of studies on prebiotics. Prebiotics are essentially sugar compounds that will nourish the good bugs. So in other words, if you take some prebiotics, what you're doing is you're nourishing the good bugs, which will, of course, multiply and colonize your GI tract. A second way is through something called probiotics, which are in fact actually ingesting a sample of good bugs. Now, in fact, you're already probably familiar with the fact that yogurt contains good bugs, and you may have, in fact, at some point been on antibiotics, and the doctor suggested that you take yogurt. And that yogurt is essentially suggested to supplement and restore your healthy gut microbiota. The third area of studies are something called FMT. And you may be wondering, what on earth is FMT? I'm going to give you a clue. Have you got it? Fecal microbial transplantation. Feces. Fecal transplantation. Or repopulation. Okay, so let's talk about what we mean by this. Let's talk about how this works. First of all, a healthy donor is selected based on a rigorous screening protocol, and they provide a stool sample. That stool sample is prepared into a slurry. That's right, looks like a chocolate milkshake. Not such a pleasant one. And then it is administered to the patient, and it's administered by either colonoscopy, enema, or, nasogastric tube. You've got it. In the nose and down into the stomach. So, FMT therapeutic applications. How has this been applied, and where has it been successful? Well, it's already been highly successful in the treatment of C. difficile infection. So C. difficile is actually a diarrhegenic infection, and it's considered an opportunistic pathogen, meaning that it only takes advantage of you when you're down. When might you encounter C. difficile infection? You may go into the hospital to have some kind of operation. You're perfectly healthy, but during or post-operation, during your recovery in hospital, you encounter C. difficile infection, and you end up with chronic diarrhea. Then you take a course of antibiotics to get rid of it, and you feel like you're cured. But then it comes back. another course of antibiotics, comes back again. These people can go on for years with chronic, miserable diarrheal disease. And one of the things that we found in the research is that their gut microbiome was severely disrupted. And fecal microbial transplantation has been used very successfully to treat these patients. Where else has it been used? Well, there are now preliminary studies to treat individuals who have inflammatory bowel disease. And there is some good promise with these results. FMT is also being experimented on to restore insulin sensitivity. And again, they're experiencing some success with this treatment. I should tell you that this is not a treatment that you could get by going to a doctor. This is considered to be an investigative new drug, and you would have to be part of a clinical trial in order to participate in this kind of a treatment. Not that I think you probably would want to. What does the future hold? Well, of course, we'd like to get away from the yuck factor. We'd also like to get away from the unknown factor in a fecal sample. So the idea would be is to create synthetic multimicrobial stool substitutes where we actually decide which bugs we need to put into this concoction, and then we use that synthetic concoction to administer to patients. And this just shows you some of the experimental results on treatment of patients with C. difficile infection using fecal microbial transplantation. And I mentioned to you earlier that there is a change in the diversity of the gut microbiome in individuals who are ill, and in fact we find that in C. difficile patients. And this bar graph here, by the different colors, shows you the composition of that gut microbiome. So you can see this is your donor sample, your donor stool sample, and you can see the colors and the proportion of those colors. Compare that with the recipient who is ill with C. difficile. Well, you can see there's a big distortion in their gut microbiome in terms of the composition. A lot more of the category of bacteria reflected by the gray portion of that bar. However, after fecal microbial transplantation, you can see that the colors and the percentages of those colors are beginning to approximate the donor sample. And in fact, that's what they're finding, is that the microbial diversity is restored. And we think that that is playing a key role in helping these individuals to recover from C. difficile infection. Okay, so what have we learned today then? Well, the first thing is we've learned that the human GI tract contains a rich, diverse set of microbial communities along its length. We've learned that the communities change along the entire length of the GI tract. We've learned that there's a key role for the gut microbiome in health and disease, and that changes in our gut microbiome are linked to a number of diseases. When we talk about the pathogens, we've learned that they use molecular cues that are encountered along the GI tract to optimize both survival and infection. And the last thing is that we now understand that the gut microbiome can actually be manipulated with diet, with prebiotics, probiotics, and with fecal microbial transplantation. And so I'd like to finish by reminding you, never underestimate the power of the stool.